everyone. I am Shreya Kamble. I refer review article Introduction to Biosensor with authors Nikhil Bhalla, Pawan Jolly. Today we are going to discuss about biosensor introduction, histological background, their components, its characteristics, application, impact biosensor, challenge in biosensing research, conclusion and reference. Let to the start the main point of the introduction biosensor. Biosensor is a device that the measure biological or chemical reaction by generating signal proportional to the concentration of any analyte in the reaction. Biosensors are employed in applications such as disease monitoring, drug discovery and detection of the pollutants, disease-causing microorganisms and marksheet are indicators of a disease. Biosensors are nowadays in biomedical diagnosis as well as wide range others such as a point of care monitoring, treatment and disease progressive, environmental monitoring, food control. Historical background. The history of biosensor dates back to as early as 1966 when young Creamer demonstrated that the concentration of an acid in a liquid to proportional to the electric potential that arises between parts of the fluid located on opposite side of a glass membrane. Between 1909 and 1992, Griptin and Nielsen first demonstrated embolization of the enzyme invertase aluminum hydroxide and charcoal. The first true biosensor was developed by Leland C. Clark 1956 for oxygen detection. He known as the father of biosensor and the invention of the oxygen electrode bear the name of Clark electrode. The main following compound is the biosensor is analyte, bioreceptor, transducer, electronics. This is a diagram of the biosensor. The first compound is the analyte. The analyte is the substance interest that need a detection. For instance, glucose is an analyte in a biosensor designed to detect glucose. The second component is a bioreceptor. Bioreceptor is a molecule that specifically recognizes that the analyte is known as a bioreceptor. Enzyme, cell, DNA and antibodies some example of bioreceptor. The third is a transducer. The transducer is the element that converts one from energy into another. In biosensor, the role of transducer is the convert to the biorecognition event in a measurable signal. The fourth component is the electronics. In electronics, this is a part of the biosensor. The process of the transduce signal and the prepare it for a display. It consists complex electronic circuit that perform signal conditioning such as the amplification and conversion signal from analogous into the digital form. The last is a display. In the display consists user interpretation systems such as liquid crystal display of the computer direct printer that generate the number of curves understood by the user. This is the overall information about the components. Then the characteristic of the biosensor. First is the reproducibility. Reproducibility is the ability to the biosensor to generate identical response for the developed experimental setup. The reproducible is a signal provide high reliability and robustness to the interference made on the response of a biosensor. Then next, the sensitivity is the second characteristic and selectivity. In this case, the minimum amount of analyte can be detected by biosensor defined as the limit detection LOD. LOD is the limit detection sensitivity. It the number of medical and environmental monitoring application biosensor is required the detect of the as a low even confirm the presence of trace analyte in a sample. The application of biosensor, drug discovery, disease detection, environmental monitoring, soil quality monitoring, food quality monitoring. Application of biosensor have a very range of wide range of application aim to improve the quality of life. The range convert their in for environmental monitoring, dissection, food safety, drug durability. This is the application of biosensor. The next, what is the impact of biosensor? Looking at the never-ending the literature related to the biosensor over the last 2 decades, reveal these biosensors are attractive not only the academic but it also industry. Biosensor technology exploit the unique prop interest of a biological recognition event on transducing device such as an event. Thus, interaction of the analytic within bioreceptor is converted into suitable output that is easily readable for the users. The next, what is the challenge in biosensing research? Biosensors have been under development for around 50 years and the research in this field has made the thermo contribution in academic over the last 10 years. Besides lateral flow pregnancy test and electrochemical glucose biosensor, very few biosensors have achieved global communication success at the retail level. There are several factors for this difficulty in translating academic research to commercial viable prototype by industry, complex regulatory issue in clinical application. The next conclusion, in vitro molecular biosensors are nowadays ubiquitous in biochemical diagnosis as well as wide range of others such as point of care, monitoring, treatment and disease progression. The biosensor device require the interaction different lines and rely on very distinct aspects such as the study of interaction of biorecognition element with biomolecule, embolization of biomolecule of the solid surface, development of anti-fouling surface chemistry, design and fabrication, and integration of the biology with the device, microfluidics on chip electronics package sampling technique. Summary, biosensors are nowadays ubiquitous in different areas healthcare. Pregnancy tests are glucose monitoring sensors have two main examples of the very successful biosensor device. So particularly for referring this content, we can further refer these lines for the additional information. Thank you.